first, I want to introduce the panelists that we have here. We have Tom Chula, who's CMO of Clearside Biomedical, Mehdi Gasmi, President and CSO at Verum, Mark Humayun's Professor of Ophthalmology at USC and founder and advisor to Regenerative Technologies, Steve Piccola, Senior Vice President and CMO at Regenix Bio, and of course Glenn Yu, an Associate Professor of Ophthalmology at UC Davis. So I'd like to start off the discussion with a kind of a broad question, and that has to do with the pros and cons of the different delivery approaches that we're studying right now for gene therapy. We have intravitreal, subretinal, and suprachoroidal, as Glenn very nicely summarized, and we have experts from all three of those approaches here. So, Mehdi, why don't we start with you, and what are your thoughts on these different approaches?
So I don't know so much about number 3, so I'm not really going to say anything about that. Intravitreal versus subretinal in gene therapy, it really has to do with what you're trying to target, right? In the case of having a gene that needs to be specifically delivered to an RPE, for example, at the back of the eye, intravitreal approach may not be the best, because, as it was mentioned, it's the difficulty of this inner limiting membrane that constitutes a major barrier to a vector that's being delivered in the vitreous to get to those RPEs that way. At least with the vectors that are currently available, which are largely what was described this morning, naturally occurring AAVs that we found in nature. Those vectors are not, they were evolutionarily evolved to transduce like airway epithelia and the gut, not so much retinal cells and to cross the LLM. So they don't do that very well. So they need to be administered subretinally. And in that case, they do a very efficient job at transducing those cells. In the case of when you want to express a protein that's going to be produced in the vitreous, for example, like a trophic factor or an anti-VEGF factor, for example, then it doesn't matter so much which cell really makes it, as long as you have as many cells as possible to make enough protein for it to be efficacious. So in that case, an intravitreal approach would work, and this is what we're practicing at Verum. So we have this new vector, which is a chimeric vector. It's not found in nature. It's been engineered out of an AAV2, which the parental vector is a natural vector, but it's been engineered to transduce a lot of cells in the intraocular space, the retina. So it doesn't transverse the ILM so much, but it can transduce very efficiently a lot of cells. When it has access to them, it does that very well. And then those cells, each of the cells, it can be in the anterior segment, it could be in the retina itself, where it has access, and each cells become a little factory at producing aflibercept or whatever protein. And then the protein is just like delivered, just like the recombinant protein being delivered in the vitreous. Then it makes its way back to the choroid, for example, or retina, where it does its anti-VEGF effect. So again, both approaches are totally valid. It's just dictated, the way to do it is dictated by the indication and what cell you really want to target. Sorry if I was a little long.
Very good. That's perfect. Thank you. Tom, thoughts?
I agree with the above, and I think the answers are obvious in terms of complications of subretinal surgery and so forth. But I'm going to look at it from a different perspective. If we look at it in terms of innovation theory, if a disruptive innovation is one that is accessible, it's inexpensive. And so you think of the computer. A mainframe computer is not disruptive. By making it small, putting it on a desktop, putting it in a laptop, putting it into your phone, it becomes accessible and it becomes disruptive. We all have computers in our pockets now. It's the same thing just with the cell phone itself. We used to, for those of us old enough, it used to be a car phone, and you used to have to go into your car and make a phone call. And now, again, we have cell phones in our pocket. So I think to make gene therapy really disruptive, we need to make it accessible. So at Spark, for example, I help, and Paulo, raise your hand, Paulo Falabella and I help set up ocular gene therapy treatment centers at Spark. We set up 8, and it's now 10 around the country. And that's a great system. You know, we had really highly trained surgeons. It was a great system for an ultra-rare disorder. We had really good control over the training. And we could deliver, we knew that we were delivering the therapy precisely to the cells that needed to be targeted. But to make it a really disruptive therapy, we need to have it accessible to patients. We need to get it into the doctor's office. We need to have it to be inexpensive. So I think ultimately it depends on what we want to do, but if we want it to be accessible, if you want it to be disruptive, for example, an anti-VEGF therapy, it needs to be something that is not at an ocular gene therapy treatment center model. It needs to be accessible to patients, in the local doctor's office.
Okay. And Steve, did you want to make any comments on this?
Sure. So we've heard about intravitreal and subretinal, and I think it's great. Tom, your work with Spark and Paulo, who we brought on board at Regenexbio earlier this year to work on our subretinal program and our other NAV technology-based AAV programs. The one thing I'd say is to step back and think of, at the end of the day, the therapies need to be effective and safe and well tolerated, that nobody wants to give up on efficacy, for example. And I think that's as true for the common diseases that we would target with gene therapy to provide a therapeutic protein. So in our case, at Regenexbio, our lead program, RGX-3114, we utilize AAV8, one of the new generation AAVs in the NAV platform. And as you heard from Ram this morning, what's interesting and the potential advantages that we see with these second generation naturally occurring AAV vectors is higher gene expression, longer gene expression, better tropism for different tissues that you are targeting. In the case of AAV8, we know that those have very high tropism for the retinal layers that you want to transfect, such as the RPE and the photoreceptors. And as Glenn elucidated, one issue is the vector, another is how do you get it there? And from our perspective, the gold standard is subretinal. That's what we know from an approved product. Luxturna definitely works. That's what we've seen. And now we have the benefit of actual clinical data with protein expression, prolonged protein expression, correlating with functional and anatomic outcomes. So I agree with all the points that have been made, and I think over time, certainly the goal is to make this approach more accessible. So knock on wood, these approaches work. We see prolonged protein expression. We see that translating into safe and effective therapies. And that will be the drive for companies and working closely with retina specialists and companies to make the procedure more standardized and even automate as much as we can to simplify the procedure to make it as accessible as possible.
I just want to add quickly, one of the things we found that's also different between the different types of injections is the inflammatory response or immune response. So we do know in general that when you give an intravitreal AAV, there's often significant intraocular inflammation that has to be suppressed with immunosuppression. And we actually noted in our experiments that for suprachoroidal, there's actually very little systemic increase in neutralizing antibodies, but there's a little bit more local inflammation. So it might help divert the type of immunosuppression, whether we do local steroids to suppress the immune system, but that I think is going to be also a major contributor.
Okay, well, very good. Let me change gears a little bit and look at things from a patient perspective. So there was a time when there was some stigmata surrounding stem cell therapy, gene therapy. You kind of alluded to that. What do you think is going to be the patient perception when they're addressing gene therapy? What percentage do you think, ballpark, will actually be receptive to this? Tom?
Yeah, this is a great question. Some of my retina colleagues were surprised when I shared some experiences from Spark. So we had done some market research and spoke to some patient advocacy groups. And it's a small percentage, but there is a distinct percentage of patients who had retinitis pigmentosa, and they were very successful and well-adjusted as adults. And they did not want genetic testing, nor did they want treatment. They had been well-adjusted, and they really didn't feel that they were disabled. Their blindness was sort of incorporated into their life. And I think, again, a lot of my colleagues have been surprised when I've shared some of this, but I also think that we're all victims of a selection bias, because the patients that come see us, and I still practice a little bit, the patients who come see us, CS because they want treatment, and the patients who seek our therapies as an industry seek our therapies obviously because they want treatment. But there are patients who we never see, and if you actually do the market research and talk to patient advocacy groups, there's a small, maybe it's 1 or 2% that are well adjusted and don't want testing or treatment.
Anybody else have any thoughts? Mark?
As far as stem cell therapies, I mean, I can give you my own experience where somebody who's lost vision clearly is very, very interested in getting that back, and stem cell therapy is something that is very intuitive to them. But the real data is California has these propositions, from those of us in California know, and about 5 years ago, they had a Proposition 71, which actually, the population of California had to vote for. And they had to get X number of votes, and it was significant. It was, not insignificant. Tens of millions of people had to vote for it, and they voted for stem cell therapy to be researched and to be developed to the tune of, $5 billion that were reserved from the taxes that are taken from us at a state level. So all that to say is, I can give you my experience, but that is very powerful. What do people think about stem cell therapy? At least in California, they voted for Proposition 71, which was significant to save and redirect tax dollars.
Very good. Okay, I'm going to move on, and I want to talk about what factors we think are going to impact the physician decision in deciding whether or not. So look at the availability that we may have with more sustained drugs that last 3 to 4 months, 6 months, 12 months, even longer with some of our delivery systems. And if and when gene therapy comes to the market, how are they going to make that decision? What factors do you think they're going to consider? What are you all thinking about addressing in their minds? Steve, do you want to start?
Sure. I think one adjective we hear a lot when we think of these less frequent injection approaches is incremental benefits. But whether you call it incremental or something else, I think it has been very impactful. And we see how the field has moved from to Eylea with less frequent injections, and in the future perhaps with brolucizumab and others that follow. I think one aspect, though, is all of these approaches still do entail indefinite treatment for the rest of these patients' lives for these VEGF-driven diseases that don't go away. So I think where gene therapy is different, if you can have sustained delivery, it's really for the first time that you can have a foundational anti-VEGF therapy on which you can overlay additional anti-VEGF treatment if needed. So the goal would be in some percentage of these patients, perhaps the less anti-VEGF needy, let's call them, that you could have a once and done treatment for these patients. But even for your tougher to treat patients, having a steady foundational anti-VEGF level in these patients, the goal would be that this will decrease the need for and the treatment burden associated with additional therapy, whatever that might be in the future.
Great. Mehdi, do you have any additional thoughts on that?
Yeah, I'm going to sound like maybe a broken record, but to me it's an indication thing. There are like genetic disorders that make people blind. Definitely the adoption should, if it's proven to work, the adoption should be no-brainer. From another perspective, I think it's going to be risk-benefit ratio. What's the safety of the approach versus the incremental as well? The product will have to be at least equal or better than what's out there, particularly when there are products on the market. So definitely the unmet need in the case of anti-VEGFs, the unmet need is there, the burden on patients, the elderly not being able to have access to treatment, and because they have other comorbidities that prevent them from getting those treatments, if they can get a one and done, as I was saying this morning, I think that would be a no-brainer.
Yeah, I think what Tom said earlier, like the word accessibility is extremely important, because I think if you have a very easy to perform with a high, efficacy compared to something that's very, very difficult and very marginal efficacy, that would be a big difference for whether doctors will adopt it. And also, when you mentioned earlier about stigmata from patient standpoint, I think there's often more stigmata from the physician standpoint. I know many local physicians who say, oh, don't worry about gene therapy, we're still far away from it. And actually the patient's the one asking for these treatments. And there are people who don't know about the advances currently, and they don't know how to do it. So then they tell the patients you don't have to worry about it.
Mm-hmm.
I think the answers are even more complicated because I think you actually have to look at the economics of the physician practice business model. As we discussed earlier today, anti-VEGFs, even though it's repetitive and a lot of doctors complain about it, it's a huge part of their business model. Doctors are now building new buildings, redesigning their offices, and really establishing, injection lanes. And if you start a one-and-done therapy to replace that, it's going to disrupt their business model enormously. And then if you ask them to refer a patient out to an ocular gene therapy treatment center for a one-time treatment, I don't think they're going to like it. And so I think that, admittedly, these are complicated diseases as we discuss, and VEGF is not the only driver. So I think it is true that VEGF, potentially a one-and-done treatment will be foundational, and we may have other therapies, the Tie2 receptor, PKI, but I think the doctors have to definitely be involved in the therapy, and we have to think about the doctor business model. So one and done may not be good for adoption.
Yeah, so there's certainly no question about that, and we hear all the time that, doctors are too busy, they don't want to see more patients, and that's just simply not true. There's no doctor that wants to be less busy or see fewer patients. They just want to figure out how they can see more better. But one thing that is happening is that this population's growing exponentially. We're going to double by 2060, and the other, obesity is increasing the diabetic stuff. So I think that some of this is going to simply help us manage that onslaught of patients rather than really hurting the physician practice. So this is really for Glenn, just a very quick question, and that is, because it's related to gene editing. Use your crystal ball 5 years from now. This is a pretty heady subject, gene editing. Where do you think that's going to be? How do you think it's going to fit in with all this other stuff we're talking about?
Glenn, he's asking for the CRISPR ball.
Right.
The CRISPR ball. I think the difficulty, with gene editing compared to other gene therapies is that it's manipulating the very genomic, your genome, which is part of your DNA, your genetic makeup. And I think the regulatory hurdle to get to that level is going to be significant because any change you make are permanent. Gene editing right now at this current stage is quite stochastic. It's very random. It cuts at a very specific location, but whether it actually knocks it out is not. So that's why there's a range of the response. And I think in terms of FDA approval, if you have something that's 30 to 50% success, that's not really a great model. So I think it still needs a lot of work, and our lab's doing a lot of that work of trying to improve the specificity, maybe a CRISPR that cuts and then stops so it doesn't keep going looking for more stuff to cut, stuff like that.
Great. So I want to shift a little bit now and talk a little bit about stem cell therapy, and I want to focus specifically on age-related macular degeneration, not other stuff. When we think about stem cell therapy, I can break it up into 4 different areas that I think are pretty distinguishing characteristics. One is who provides the cells. 2, the RPE cell origin. So are they embryonic or are they induced pluripotent stem cells? Is it being delivered as a RPE cell suspension or as a monolayer being injected under the retina? And then when you actually look at those monolayer implants, what is the monolayer substrate? And there are different types. And so I want to kind of hit on these topics very quickly. And starting with Mark, let's talk a little bit about who provides the cells and how does a research organization decide whether they're going to use autologous cells or allogenic cells? And I think that has something to do with immunogenicity. There's scaling issues that come into play. What's your impression of that?
Well, yeah, I think you've correctly outlined autologous versus allogeneic. With autologous coming from person receiving the transplant, you don't have to immunosuppress. but you're still dealing with potentially the same genetic defect that led to the disease. As Glenn pointed out, allogeneic comes from a match-related or unrelated donor, as you point out, so they do need a little bit of immune suppression. But for the eye, you know, one of the interesting things is that you could potentially do that very locally with a steroid implant or something like that in the eye, and you don't have to do systemic therapy. So for the eye, that's a game changer. Maybe that'll work out and you don't have to do immune suppression.
Do you think it matters if somebody's evaluating companies and they're trying to figure out, okay, there's all these variables, all these forks in the road that people can take when they're developing this, is it better to be looking at a company that's using autologous or allogenic? Or does it not matter because there's different ways to manage it?
Yeah, I think it, you know, just depends on how well it works. And Emmett, we were just talking earlier, and why am I the only guy up here who really does stem cell work per se? And that's because, you know, we need a win. And once you get a Luxturna type win, then there's a lot of interest in the stem cell area. We need that first company to break through, and then I think there'll be a lot of interest. But to answer your question, I think it'll just depend on how well it works.
Okay. And what about RPE cell origin? And anybody can take this. You know, we have the human embryonic stem cells and then the induced pluripotent stem cells. There are different factors that impact the use of both of those, and how should people look at those, or should they look at those equally and not worry about it?
Glenn, you want to give it a shot?
I don't do any stem cell work myself, but I mean, ultimately, I think the ethical question's going to come because I feel like, especially with you know, abortions and use of embryonic tissue, it's so charged right now. Actually, I have an NIH grant, and apparently, because I had human fetal RPE in my grant, there are now like all these levels of oversight and review that is questioning whether I can continue without it. I think that that's going to be a potential issue compared to iPSCs. Tom?
I think iPSCs, I always marvel at this. The idea, I think it was in 2006, Yamanaka and colleagues came up with the idea of from a mouse fibroblast to put transcription factors on mouse fibroblasts and coax them back into an embryonic state. I just think that's so amazing. It goes back to the discussion we were having earlier about lasering people's fovea 25 years ago, because that's what you're supposed to do according to the macular photocoagulation study. And it's amazing how far we've come. But I think the problem with induced pluripotent stem cells is that it's extremely expensive to make an iPSC for every patient. It's kind of like on the scale of CAR T. It would be that expensive. And you know, in Japan, they're actually making them and HLA typing them so you could literally just take them off the shelf potentially. So you could HLA type a patient and pull off a induced pluripotent stem cell. It's already HLA matched to the patient. So that's a way to mitigate against some of these expense issues and mitigate against some of the ethical issues. So it might be a path forward potentially.
Yeah, very good. So I want to kind of, again, jump ahead, and I want to move now to the different implant procedures. So injecting a suspension of cells versus a monolayer. And can we switch to my slides off of the video here? Very good. And so this is some research by Takahashi from the Ripken Institute in 2017, and they created these allogenic stem cells, which is really a remarkable thing because now you can have something that's prepared independent of the process and have it on a shelf and ready to go. And so they injected this into the subretinal space, and their belief is that the cytokines caused a regression of CNV. And so, one, I want to hear a quick comment about whether or not you think that suspension of RPE cells under the retina is a viable approach. Obviously, there's not a lot of research on this, but just your gut feeling.
Well, the problem about RPE is that they're extremely polarized. So the top of the RPE is completely different from the bottom of the RPE, the apical and the basal side. All the molecules expressed are very different. So I don't think a suspension, unless that there is some kind of way to orient them. Even when we try to collect RPE cells, it's very hard to scrape them and then try to regrow them as a sheet. It's actually almost easier to have little fragments of sheets and try to grow it that way. Mm-hmm. So I think it's hard with the suspension. And
so this brings us to the really interesting stuff that Mark's group on the left looked at these RPE cell sheets on a Paraline membrane, and Lyndon de Cruz in London with the London Blindness Project was looking at it for neovascular AMD. So Mark's group, dry AMD, Lyndon's group, neovascular, but they would both apply to both. And it's interesting, when I look at this, you know, I think it reminds me of that old Navy principle of keep it simple, stupid. And I look at the subretinal injections, and we're all very accustomed to doing those, and it's very simple. And then you look at Mark's implant, and it's, you know, extremely, extremely complex. And that's, you know, the guy on one shoulder, and then there's the guy on the other shoulder going, well, you know, Mark did not win the National Medal for Technology and Innovation by keeping it simple with the Argus implant for RP. And so, Mark, I want to, actually we're going to skip that, and I want you to talk a little bit about the different membranes, the substrates that these go on. So you use a parylene membrane, Linden uses a polyester non-diffusible membrane, and then out of NEI they have a biopolymer. How do we look at these?
First of all, the simple thing. So there was a wise man who once said, keep it as simple as possible, but no simpler. And that was Albert Einstein. So you got to address what you're dealing with and not oversimplify it. So you got to make it as simple as possible, but not any simpler than that. So with that, you know, the suspensions, as Glenn just pointed out, those cells really need to be oriented to produce the type of growth factors they need to, and to really do the phototransduction. Now, you may be able to inject some, and they may take a hold and may form a couple layers, you know, 2 cells or 3 cells that are oriented properly. But I think orientation is going to be really key here. You know, then it takes you to these different types of substrates, and each one has their issues. So if you take an erodible substrate just for a second, how is that eroded? And you know things are eroded in the body because macrophages come in and they gobble up the biopolymers, and that sets up an immune reaction. And then secondly, when should it go away? Should it go away in 3 weeks, 6 weeks, 8 weeks, 12 weeks? So it's very hard to get it, what I call the Goldilocks principle, just right to have it you know, go away at the right time. So because of those things, it was too difficult. We spent 5 years on it. We couldn't develop an absorbable approach. We went to a non-absorbable approach, which is the parylene. Polyester is sort of in that range. But then the second constraint is the diffusion constant from the choroid to the retina. You can't block that, or to the RPE. And polyester is very difficult to work with. It creates inflammatory scarring, and it's very difficult to control the non-diffusible aspects of it. So we basically used a room temperature CVD monomer deposition of parylene that we etched, just like computer chips are made, and so we can make 0.4 micron thick in different areas, and we measured the diffusion very carefully, and it allows 250 kilodalton diffusion, which is similar to Brooks membrane. So all that gobbledygook and very fast technical talk to tell you how we ended up on parylene.
Yeah, if I was in your shoes, I would have used that room temperature CVD thing too. Yeah.
Okay.
Okay, well, I think that's the last word. We've used up our time. Thank you all very much. Thank you to our wonderful panelists.